Hello, and welcome back to Living in Water. Today we will be doing part 2 of this lecture. In the previous lecture, we discussed ventilation, buoyancy, and senses as related to aquatic vertebrates. And today we're going to continue this discussion looking at the internal environment and temperature. Water is very important to aquatic organisms, and we're talking internally now, not externally. So almost everything that happens in the body tissues of vertebrates involves water. The maintenance of that water and solute concentration is vital to survival. And you can think of this in terms of being dehydrated, right? When your water levels become low, it's kind of important to your survival to bring those water levels back up. The temperature for these reactions is also important. So when it comes to maintaining the solute level in your body, the kidneys are very important for this. So they remove the waste products from our blood. The blood gets filtered through your glomerulus. The glomerulus are in your kidneys, in your nephrons. So each kidney has millions of nephrons in it, and those nephrons have the glomerulus, and the glomerulus is where the filtration takes place. And this is unique to adult vertebrates. This ultrafiltrate is processed to remove the water and essential metabolites. So these are the things we want to keep in our body, like the glucose and the amino acids, leaving the urine behind. So our kidneys are very efficient at pulling out everything they possibly can that we want to keep in our bodies and leaving just the waste behind. So we do have the regulation of ions and body fluids. And this uses some of the things we've learned from chemistry, right? So this hopefully is a little bit of a review. So we have the idea of osmosis. If you have a lot of particles concentrated on the outside of a barrier and fewer on the inside, those are going to try to flow through. The particles will flow through until we're equal on either side. And this happens passively with no direct energy spent, just a simple osmosis. We can have facilitated diffusion, where we have specific channels that help move the particles in, those ions back and forth. We can have a carrier protein that again helps pick up those particles and move them through across that membrane, dividing it. And the last one we have is active transport. So active transport is when we're moving the particles in, but we're using a protein pump to do this. And this uses ATP, and this is the important part here. This uses energy to fight the gradient, to bring particles where we want them to be, not where by osmosis they would like to be. This will make a little more sense in a minute. So here we have a chart full of all sorts of scary looking numbers and words and things, and it's not as bad as it looks. On the left-hand side, this is the type of the animal that we're going to be looking at. In the next column is the osmolality, and that's just kind of telling you how much stuff is in its blood, how many particles are in there. And you can look specifically at the things like the sodium levels and the calcium level, or the chloride levels that are present in the blood. So what we're going to do is just take a subset of this to look at to start with. So to start, at the top of this chart, we have the two different types of water. So we've got seawater and we have fresh water. And as we'd expect, seawater has a much higher osmolality than freshwater does. Anyone who's gone swimming in one of the oceans around here, gotten some water in their mouth, you can taste that salt. It's present in high levels. If you swim in freshwater, you're in a river or one of the lakes, you see that there's not as much stuff, right? Our osmolality is very low, and that makes sense. There's not a lot of sodium or chloride in there. We don't taste that salt when we're in the water. Now we'll look at some marine animals with this. So marine invertebrates, their osmolality is 1,000, so it's right around the same thing as seawater. They're not fighting any sort of balance. Their body tissues, their blood levels, the concentrates of the ions in their blood is approximately the same as the seawater surrounding them, so there's no fight for the water or solute balance. When we look at the marine vertebrates, so our hagfishes, remember our hagfishes, that was kind of one of our basal groups, they're right around the 1,000 as well, suggesting that their body tissue is the same way, showing kind of a similarity with invertebrates here, but basically they don't have to worry about any sort of solute balance gradient either. Teleosts, on the other hand, we see is a lot lower. It's less than 350, so they're going to be wanting to keep the salts out, keep their water in, and they're going to be fighting against the current, the gradient between the seawater. Bull sharks, well, those are pretty close, but they're a little above, so they're also going to have a gradient that they're going to be looking at. So if you look at the teleosts here with the less than 350 osmolality compared to the seawater they're in, where it's around the 1,000, We call that a hypoosmotic. They have a lower solute concentration in them than we find in the surrounding medium. Those bull sharks are opposite of this. They have a higher concentration. They are called hyperosmotic, a higher solute concentration than the surrounding medium. All right, so now we're going to switch this and we'll look at the freshwater. So we switch out those animals on the bottom, and now we're looking at the freshwater vertebrates. You see we have the teleost, so those are our bony fishes. Our bull sharks are sharks that can go back and forth between the two. We have a freshwater sawfish, and we have amphibians. And you can see that their numbers are all well above the 10 that we have in freshwater. So now all of these organisms have a higher solute concentration than the surrounding medium, which means they are all hyperosmotic. So we have these two basic scenarios here when we're talking about fish. So a marine fish lives in its environment. There's water and there's salt. The salt outside is trying to get into the fish because the fish is less concentrated than the salt. In the same vein, the fish's water is trying to go out, to diffuse out, to make it equal along either side of the membrane, right? So that the fish is fighting the idea of being equal to the outside environment. The freshwater fish, this is the opposite, right? In this case, its salts want to go out and more water wants to come in because this fish is more concentrated than the external environment. So one of these is hyperosmotic, having a higher concentration inside than the surrounding medium. And one of these is hypoosmotic, having a lower solute concentration than the surrounding medium. And the question is, which is which? Who gets the blue arrow? Who gets the red arrow? Give you a minute to think about it, and we'll see if you can pick right. All right, hyperosmotic, that's going to be the freshwater fish. These fishes have more solutes inside them than are in their outside environment. All right, so now the hypoosmotic, everybody better get this one right. That's going to go to the marine fishes because they have a lower solute concentration than the surrounding medium. Does that make sense? I hope it does. As we saw in the chart, there are some organisms that we find on both sides in the marine as well as the freshwater, but most organisms, most vertebrates, have a difficulty surviving a range of salinities. So most are going to fall into the stenohaline category, steno being narrow and haline being salt. So they can tolerate minor changes in salinity. You couldn't take a saltwater fish usually and move it into a freshwater lake and have it survive. Euryhaline, uri being wide and haline being salt, these are vertebrates that can tolerate a large change in salinity. And we have bull sharks. They can do this. They are one of the few that are able to go from saltwater to freshwater. The Jaws shark was based on bull sharks that have that ability to be found in rivers as well as in the ocean. When the shark moves between those two environments, the salt and water gradients completely reverse with the movement between those. So we do see some specializations that we'll get into in a minute. We'll start though by looking at some freshwater vertebrates. So we've got two main groups here. We've got our teleosts and we have amphibians. So teleosts, again, those are our bony fishes. Teleosts' main issue is that their gills are permeable. So water can flow in and out and ions can flow in and out. So it's this permeable membrane that they kind of have to battle with. So as they breathe, right, water flows over the gills to get the oxygen, and a lot of that water is absorbed to come into the fish because these fishes are more concentrated than outside, so the water flows on that gradient. So these fish don't have to drink water because the water is constantly flowing in across the gills. Because there's so much water they have to process, when we look at their kidneys, we see that they have these large glomeruli because they have to process all that water. That means they're going to produce a lot of urine, so they're going to be peeing all the time too. And the salts that are present in that filtrate are going to be actively reabsorbed by the fish. The gills also have ionocytes, which are sites that allow them to actively take up ions. They'll take up the chlorine, and as chlorine is negative, it's going to pull that sodium along with it. One important thing to note here is where we say actively, these are things that involve ATP, so this involves energy in order to do this. So here's kind of a little picture of the freshwater fish, and we can look at this and see all of those characters that we mentioned. So this here is the gill membrane right here, and as the fish is breathing, the water is going to go across it. and there's salts present as well. Anything that has the open arrow is going to be passive flow. So the water's passively going inside the fish because it is less concentrated outside. So the water wants to flow into the fish to make the fish less concentrated. The fish is using those ionocytes in its gills to pump in chlorine. With the chlorine comes the sodium behind it. So those are actively being pumped in. Remember, this fish does not have to drink because all of the water is passively moving in. All of this water that comes in has to be dealt with by the kidneys, and it means the kidneys are working a lot and they're producing a lot of dilute urine throughout the day. Inside the kidney here, those salts are going to be reabsorbed, actively reabsorbed by the fish as well. And then this up here, this is sometimes confusing to students, where we see the water bouncing off the fish and the salt bouncing off the fish. And all this is telling you is that the skin is impermeable. So there's no gas exchange, no ion exchange through the skin of the fish, right? The only place we're getting this is across our permeable gill membranes. Water cannot flow in, salt cannot flow in, salt cannot flow out, water cannot flow out. All right, so hopefully this little picture helps explain some of what we just talked about on the previous slide. The other major group of vertebrates in freshwater are the amphibians. They're a little different. Instead of it just being the gill membranes where we have this water diffusion ion movement happening, their entire body surface is involved in the uptake of ions. Their body surface is permeable. So they're also not going to drink. There's no need to drink. That water is diffusing through their skin. But they're also losing ions by diffusion because the water, right, through osmosis, the water and the salamander want to be the same. So they're trying to decrease the concentration of that salamander. So he's losing ions and taking up water passively. He does have, just like we saw in the fishes, specialized cells that allow him to actively take up ions to kind of work out the balance he has. This ability to take up the ions is inhibited by acidity. So if you are a freshwater amphibian and you live in an area that has high levels of acid rain or other reasons to have high levels of acid, this could be something like mine runoff, anything that would add to the water that you're in and increase the acid level, then you're going to not be able to take up ions as well, and this can actually kill the freshwater vertebrates. It's kind of the opposite of being dehydrated from a human point of view, right? At this point, you can't get the ions in, so you're actually overhydrating yourself. You have too much water and not enough salts present. All right, so we'll leave the freshwater behind, and now we'll come back to the marine side a little bit. So now we've flipped, right, what we're doing. We want to keep the water in and get the excess ions out. And we have three major groups here. We have our teleosts, again, those bony fishes. We have our hagfishes, coelacanths, and cartilaginous fishes that we kind of all just group together. And then we have the amphibians. And remember that we are dealing with hypoosmotal ones, lower solute concentration than the surrounding medium. So you can see it here again in that cartoon, right, with the fish. Here, the marine fish, those excess ions want to get in, and the fish's water wants to get out, and the fish has to fight against these gradients. So with teleosts, this is similar to what we saw in the freshwater environments. These are the bony fishes, so most of that ion movement is across those gills. In this case, the glomeruli are small and the filtration is low, and we're not producing very much urine at all because we're not drinking the water. We're not having fresh water coming in. We're actually drinking seawater as much as we possibly can. And this is kind of the weird part. The sodium and chloride ions are absorbed in the gut on purpose. Even though this fish is salty enough, it's absorbing more salt. And that's because if it puts the salt into the gut, then it makes the gradient so that that water will now flow out via osmosis into the blood. These fishes also have those ion pumps that allow them to actively pump out the chlorine ions. Actively, again, remember that's using ATP and energy. For some fishes, this could be up to 1/4 of their daily energy use. So now we've got the little cartoon picture of the marine teleost to look at. So just like we saw before, right, we've got that skin that is impermeable, no salt and water gets through, no salt and water gets out through the skin. We have our main place where we see the movement, and that's here on the gills. In this case, it's the water that passively goes out and the ions that are actively pumped out here on the gills. We are drinking lots of seawater in. The water will passively flow in because we are absorbing the salts in the stomach. So that makes us more concentrated, and that has the water flow in, and then we take the excess ions here and we pump them out. That makes sense? See how it's a little bit different than what we saw in the freshwater fish? We drink the water here, the other fish didn't drink. We're still actively pumping to move things around to where we want them to be. Hagfishes, coelacanths, and cartilaginous fishes, they're a little different. So they actually keep compounds in their blood so that they're slightly hyperosmotal to the seawater. So they keep themselves concentrated, their blood concentration, a little higher than the seawater, then they don't have to fight that big balance anymore. They do have large glomeruli in their kidney, giving them a high filtration rate. And their gills have a fairly low permeability to ions. They don't have those salt excreting cells in the gills that we saw in the teleosts, but they do have rectal glands that will allow them to excrete excess ions if they need to be. Sharks can go back and forth, like we said. There's our little bull shark, or not so little bull shark. About 5% of cartilaginous fishes are euryhaline and able to tolerate that large range of salinities. Bull sharks are one of those. So when they're in marine, they act like a shark. They have a high urea level in their blood. So they keep a lot of solutes in their blood so that they don't have to fight the balance across. When they move into freshwater, they lower that. So they're going to have to be dealing with excess water and excess ion flow and figure out how to balance that. But they have the ability to change the level of urea that's in their blood. True freshwater sharks, there are about 5 poorly known species of sharks and a few stingrays. It's a spear-toothed shark down here in the picture that are truly freshwater cartilaginous fish. And they're found in brackish freshwaters in Asia and Australia. The salt concentrations in their blood is lower than what we see in bull sharks, but it's slightly higher than what we see in teleosts. So if they were like the typical marine sharks, they would produce the urea in their blood. And they do have the enzymes to produce the urea, but if you take a freshwater shark and you put it in salt water, it doesn't just automatically switch and start doing that. That's because they've evolved to be in freshwater. They've been in freshwater for tens of millions of years, and that's led to a change in their physiology. So their gill permeability has changed and their kidney functions have changed to be able to live in a freshwater environment, not a saltwater environment. We do have amphibians that live in seawater, and this just is mind-boggling to me. This is in southeastern Asia into China. We have these crab-eating frogs that are found in intertidal mudflats. Remember, amphibians absorb salts and water over their whole skin. So basically, you're taking a sponge and sticking it out there, and it's going to be absorbing salt all the time. And how do they deal with that? Well, they're a shark, pretty much. They act like a shark. The adults will retain urea in their blood. They'll release it internally and keep it in their blood, and that makes them closer to the salt water so that when they're in this 80% seawater at high tide, that they're not fighting that ion balance as much. The skin is also permeable, which means that urea that we're making, that's going to be diffusing out as well. So they have to be able to synthesize urea at a high rate to replace the urea that they're being lost. And then the interesting thing is this is what the adults do. The adults are pretty much a shark. But the tadpoles, they don't have the ability to synthesize urea. They don't have that enzyme. So the tadpoles actually act like a saltwater fish. They use salt excreting cells in their gills to actively pump out the ions as they're diffusing in. So it's just amazing that you have these tadpoles that act like a saltwater fish, and you have these adults that act like a shark, and they're able to exploit this environment where we don't usually see amphibians. Okay, so that's how aquatic vertebrates deal with kind of their environmental solute water levels. But we also have to deal with excretion, waste products that are produced from what we eat. So we ingest food, which is basically proteins and nucleic acids that contain nitrogen. And nitrogen is what we have to figure out how to get rid of. Nitrogen gets reduced enzymatically to ammonia, which is good, except ammonia is toxic. It is water soluble and will easily diffuse, but you can't really hold on to it for a long period of time. So you need to get rid of that ammonia very quickly. So we see a variety in the nitrogenous waste produced by the different groups of vertebrates. So the bony fishes, they're in the water, so they just do ammonia. It doesn't matter if it's toxic. It's easily soluble, and they're surrounded by water. So dilution is the solution to pollution, and the ammonia comes out, dissolves. They don't worry about it. Mammals, on the other hand, and this is us, we're a mainly terrestrial group, so we don't really like ammonia. It's toxic, it's there, it's not something we want to have around. So we convert it into urea, which is not as toxic as ammonia. So we can concentrate it, and that allows us to conserve water. As a terrestrial organism, that conservation of water is important to us. And we see something similar when we look at reptiles and birds. They convert that ammonia into uric acid, which can also be concentrated. It makes that lovely semi-solid paste that we're all familiar with on our windshields, but allows them to have that water conservation as well. All right, so that got us through the internal environment. We've got one last section to talk about here, and we'll be done with this chapter. So we'll get into the discussion of temperature. So we are a leaky bag of water, and we're basically a leaky bag of water that's catalyzed by different enzymes. It's the rates of those chemical reactions that happen in there. So for those enzymes to work and do what they want to do, what they're supposed to do, it's going to change as the temperature changes. So temperature becomes important to us from a very cellular level. But also, as your energy need increases, maybe because it's colder outside, then your food has to also increase. So there's a lot of ways that temperature is important to aquatic organisms. So we're in the water, and we've talked about this before. It's just hard to control your body temperature in the water. Heat moves really rapidly. This is that idea that you go to the beach on a nice hot day and you go in the water and it's warm, and after 20 minutes or so, you've got goosebumps and you're cold, because the water is very good at leaching that heat out of your body. So you need some kind of thermal regulatory mechanism to maintain a different body temperature different than what you're in the surroundings. So we'll go for two general temperature categories here. We'll go for one called poikilothermy and one called homeothermy. So the idea here is that poikilothermy, poikilo is variable and therm is heat. So this is a variable heat. So your body temperature changes. So this would be things like fish, amphibians, reptiles, right? That when it's hot, you're hot. When it's cold outside, you're also cold. And we'll compare this to a different group called homeothermy. So the homeos are the same. Therm is heat, so this is the same temperature. So this is more like birds and mammals that have more of a stable body temperature that doesn't move that much. That makes sense? Well, it almost makes sense, because there's always an exception. Well, what about this cute little guy here? He's an alpine marmot, and his body temperature will drop from what it is normally during the day, 40 degrees Celsius, that's about 104 degrees Fahrenheit. And during hibernation, it'll drop to 5 degrees Celsius. That's 41 degrees Fahrenheit, 104 to 41. That is not homeothermy at all for a little fuzzy mammal. That is highly variable. And then on the other side, what about those tropical fish that are in fairly stable water? If you are a tropical fish near the equator, your water temperature is only going to vary by a handful of degrees throughout the year. So that makes you the stable homeotherm, and the marmot mammal up there the unstable poikilothermy. So perhaps we need some better categories. Like these, ectothermy and endothermy. So if we think of ecto as being outside, then you're getting your heat from an outside source. And endothermy, with endo being inside, you're getting your heat from an inside source. You're using those metabolic processes to create heat. And this makes a little bit better sense. So you can think of it as the endotherms as being those typically hot-blooded, warm-blooded birds and mammals, and the ectotherms as being those traditionally cold-blooded animals, the fish and amphibians and reptiles. But these are not mutually exclusive categories. Birds like the roadrunner here, they're able to save energy by cooling their body temperatures a few degrees at night. So instead of maybe being at 40 degrees at night, they'll let their body temperature drop to 38. And then in the morning, they'll go out in the sun, they'll spread their wings like this and warm up, bring their body temperature back up to 40. So in that case, it's getting heat from an outside source and acting like an ectotherm. And over here on the other side, we have this python, which we tend to think traditionally as being an ectotherm, right? It's the cold-blooded reptiles. But what these guys will do, or I should say these women, because this would be the female, they'll wrap themselves around the eggs and they'll contract their muscles. So anyone who's contracted their muscles, right, you do some exercise, you create heat. And by doing this, to the eggs. They're generating heat, and now they're acting like an endotherm, and they're using this heat to incubate their eggs. So if they can brood their eggs at that higher temperature, they're going to hatch a little bit bigger and a little bit faster than otherwise, and maybe give the young some kind of advantage. So you can see that they're not mutually exclusive. We would generally say that the bird here is an endotherm, even though it shows some ectothermic properties, and we would generally say that the snake over here on the other side is going to be an ectotherm, even though it's showing endothermic properties. And then just to confuse things even more, we will have what's known as regional heterothermy. So basically hetero here is different, right? So this is just the idea that we have different temperatures in different regions, different parts of our body. Some fishes can have parts that are 15 degrees Celsius warmer than the water. That's almost 60 degrees Fahrenheit. So this allows some fish to exploit certain habitats. If you're a warmer fish, you're going to be a faster fish. If you're a faster fish, you can catch that prey better. So what we see are these warm muscles. So we're going to go back to another Redia mirabilia. In this case, it's that wonderful net, right? All of those veins and arteries in there. And they retain the heat produced by active muscles. So again, it's that idea that when we use our muscles, we produce heat, right? You go out for a run, you do a couple deep knee bends, you're going to feel the heat, you're going to get hot. And that's because when our body converts the glucose into energy, right, we lose, it's not very efficient, and we lose a lot of as heat, and the fish are able to take advantage of this heat. So the cold arterial blood from the gills will flow through this net, and it's going to be warmed by the warm blood that's coming out of the tissues. So you use the muscles and produce the heat, and you have warm blood, and that cold blood comes in from your gills and it passes across that warm tissue. Now you're warming up that blood that's coming in. This can help fishes keep their muscles 5 to 10 degrees Celsius warmer than the water around them. Again, warmer muscles, faster fish, better predator. We see some really cool examples of the heterothermy, the regional heterothermy. Here we have hot eyes. So these we see in the billfishes. Their brain and the eyes are warmed by modified eye muscles. And in this case, they're producing the heat on that cellular level by cycling the calcium. And the deep divers, those that go deeper, so they're going to be in the colder water, they have better developed heater organs than those that don't dive as deep. And then we see this as independently arose 3 times in the billfishes. So by protecting these parts of your bodies, your brain and your eyes, those are warmer. It's going to make you a better hunter at those deep, cold depths. And in 2015, we discovered this endothermic fish. Well, we didn't actually discover the fish. We've known about the opa. You can see down here, he gets quite big. He's also called the moonfish, and it's not that we didn't know about the fish, it's just we realized he's endothermic. So when it goes through the water, it flaps its little pectoral fins. You can think almost like a little bird flapping away, and that allows it to have this heat, right? You use those muscles, it generates heat, and the fish can use this to keep its muscles about 7 to 9 degrees warmer than the surrounding water. And it's found at depth again. So if you're down there in the deep water where it's cold and you're out there hunting those fish and the squid, having the warm body, the ability to keep the warm blood inside your body makes those muscles work faster and makes you a better fisher, better hunter. Fisherman as a fish. That's the opa. And then we also have the marine mammals. You have to remember we're in that aquatic vertebrate group as well. Well, mammals are. We are endothermic, right? So that means we produce heat from the inside. The main issue with fish is that the cold water on the gills, right? In order to breathe, we're getting cold. Every time we breathe in, we're getting cold water going across our gills. That's not an issue when we have lungs, but we still have to have some kind of insulation from that cold water. So we see this a couple different ways. We can have a pelage, that's a nice fur coat like here in the otter. It can be water repellent because we want to keep that cold water away from our skin. But in order to be water repellent, that means it has to be maintained. So you can see the otter here grooming himself to keep the oils on the surface, to keep that water on the outside, furry coat, warm, happy otter. Cetaceans and pinnipeds. So the pinnipeds, like this one here, those are our seals and our sea lions. Our cetaceans, those are the dolphins and the whales. They use blubber for insulation. So basically they have that nice thick coat on the outside of them to keep the rest of themselves warm on the inside. Marine mammals can also use their flippers to control their body temperature. So this is the idea that this is a large surface area that goes out in the water, and you can cool it by routing the hot blood. So as the blood moves out to the tip of the flipper and then back in, we can transfer the warm arterial blood over to keep the heat inside the flipper, so that when we get the ice water, it's just hitting the end. We're not wasting all of that heat, unless we're hot and we want to cool off, in which case then the veins and arteries are going to direct the blood flow so that the hot blood comes out here, hits the cool water, cools off, and brings cooler blood back into the body. So they can use it either way, whether they want to retain that heat or dissipate some of that heat. And this brings us to the idea of the surface area to volume ratio. The idea of how much surface area you have exposed can dictate your interactions with the environment. So if we start with a cube, we'll just pretend this is a little cube mouse here, and it's 1 centimeter squared on each surface, each direction, right? So the area of one side is going to be 1 square centimeter. The total surface area, right, we've got 6 sides, that's 6 square centimeters. Our volume is 1 cubic centimeter, so our surface to volume ratio is going to be 6:1. So now if we take that little mouse, and instead of being 1 centimeter, now we'll make it bigger. It'll be 5 centimeters on each side. We see that now our area of each side is 25, our total surface area is 150, our total volume is 125, and we see this huge reduction in our surface to volume ratio. And if we make that little mouse even bigger, so now we're up to 10 centimeters, we can see that now we've changed dramatically. Now we're 0.6, so we're a much larger organism, but the amount of our body that's exposed to the outside is proportionally less. And this comes down to this basic idea, this is the important part, don't worry about the numbers, that bigger species exchange energy with the environment less rapidly than small species. Okay, and this brings us to an idea of gigantothermy. So this is the ability of an extremely large animal to maintain a constant and relatively high body temperature just due to its large surface to volume ratio. So we see this we're pretty sure in those large dinosaurs, this is the large sauropods, that just because of their size, they were able to generate enough body heat that they would be considered to be an endotherm. We see this in elephants playing in the snow. It's not that the poor elephants that live in the African savanna are freezing. They're so big, they don't even know it's cold outside. And also our sea turtles over here, right? Our sea turtles can swim in Arctic waters, keeping their body temperatures 18 degrees Celsius warmer than the environment, which is pretty amazing. When you've got a big animal, it takes a long time for it to heat up, like camels in the desert. They can wander around for a long time before they get hot. And as we'll see later in the semester, those small desert rodents, they've got to have a burrow to escape into because they're so tiny, they heat up quickly. But it also has the other side where it can be difficult to cool. So if you're a tropical tortoise and you can't get to the shade, you're going to overheat, because once you're warm, it takes you a long time for your body temperature to come back down as well. So that wraps up this chapter, our living in water. We finished off the external and the internal environment today, as well as temperature. If you have any questions, please send me an email and let me know. Thank you.